Marker-assisted selection uses modern genetics to speed a traditional process of artificial selection. Artificial selection is human choices shaping plant genetics. So plant breeding, artificial selection by another name, begins with natural genetic variation between individuals. Simply by the power of choice, plant breeders can shape a variety. In this case, the naturally occurring variety can hold its breath or survive underwater for up to 2 weeks. That's the desirable trait, but a lot of undesirable ones too. Next, plant breeders make crosses between different plants with desirable traits and choose the offspring with the best combinations. In the case of rice, pollination occurs inside of a closed plant, and so here you can see a scientist making a cross by opening up the flower and using tweezers to physically pollinate one plant to another. In the case of SCUBA rice, a plant with desirable characteristics like yield, right, is crossed to a traditional plant with the desirable trait, right, the SCUBA, the ability to hold its breath underwater, resulting in a big population with thousands of plants that needed to be screened. For SCUBA rice, this meant growing the plants for months, submerging them underwater for weeks in order to find those that could survive. Then they had to be grown to maturity in order to evaluate for traits like yield. So this conventional plant breeding approach, slow but usually reliable, never really worked for SCUBA rice because there were too many negative characteristics that came along with that SCUBA gene. Once plant breeders knew the location of the SCUBA gene, though, a whole new approach opened up. They could screen the DNA directly. After making the same sort of cross, instead of screening the plants underwater, right, they could now screen the DNA. A faster process that takes a lot less space. But how do you screen DNA, which is something you can't actually see? So geneticists use markers, known pieces of DNA, like bookmarks, in order to tell them about genes. There are lots and lots of different kinds of markers, and really any identifiable feature of DNA can be used. So what this alphabet soup of marker types have in common is that they can reliably be used to detect genetic difference. So here are the 12 chromosomes of rice. Right? The sub1 gene on chromosome 9 is the SCUBA gene. Right? Plant breeders needed markers on either side of the gene in order to be able to know whether or not they'd moved the gene. So they used microsatellite markers that were different, polymorphic, between the two parents. A polymorphic marker allows the plant breeder to tell which parent the DNA came from. In this case, the donor parent has the SCUBA gene and the allele at the marker marked in blue. The recipient parent has a different allele there. Marker-assisted selection starts off in much the same way. A hole punch-sized sample is taken from a 10-day-old seedling. That's plenty. The plant can keep growing. The scientist then can extract the DNA using soap and grinding up the tissue, other chemicals as well, in order to isolate the DNA, which then they use a process called PCR, or polymerase chain reaction, to amplify the marker area. In essence, making millions of copies, enough to be visible to the naked eye. Then you have to run the DNA through a gel, like Jell-O, using electrical current to separate them by size. The result, after illuminating the DNA, is that you can see different bands for different sized markers. So in the case, in our cross, which of these, here's a cartoon of the same sort of gel, which of these plants would you use as the parent for your next cross? Well, you want to make sure that you don't want the white DNA, right? And you do want the blue DNA, and so these are the parents you would use. The donor parent, called FR13A, had lots of undesirable genes and really only one desirable one, the sub1A gene. The end product then would have really only the sub-1 gene from the donor parent and everything else from the high-yielding, great-tasting recipient parent. But that's not what an F2 parent looks like. A parent in the segregating generation has roughly 50% of its DNA from each parent. So it might look more like this. With the sub-1 gene, the parents inherit a lot of unwanted genes from the donor parent. These genes were the reason that traditional plant breeding approaches hadn't worked. But what if plant breeders could achieve something much closer to the ideal genotype using markers again? Traditionally, when plant breeders want to move only a small amount of DNA, they use a back cross. It starts with the same sort of cross between a recipient parent with good characteristics and a donor parent with something desirable that the recipient lacks. As before, the hybrid or F1 generation has equal amounts of DNA from both parents. The parent one was a popular variety called Swarna. The donor parent had the SCUBA gene. Then plant breeders make a cross back to parent one, or the recipient, or the recurrent parent. This back cross, right, results in about 25% of the donor DNA. At the same time, though, plant breeders have to screen to make sure that they're getting the SCUBA gene as they do this. Plant breeders can repeat this process. With each generation, they halve the amount of undesirable DNA and increase the proportion of the desirable amount DNA. So where do the markers come in? Instead of just two markers, plant breeders now need many, spread across the whole genome. Recall that after 2 generations of back crossing, a plant contains about 12.5% of the donor parent material. But that's an average. There's a lot of variation between plants, because where crossovers occur during meiosis is really quite random. Some plants will contain more or less donor DNA plant material. So plant breeders can use the markers to select the plants with the least amount of donor DNA aside from the sub-1 gene. The end result? Well, plant breeders were able to use a combination of traditional crosses and markers to screen for the SCUBA gene and screen against the donor DNA in other places. After just 3 generations of back crossing and marker-assisted screening, the resulting genotype had only a small piece, about 2.9 megabases, million base pairs of donor DNA on chromosome 9, and hardly anything else from the donor parent. Really pretty close to the ideal. So what this means is that by using markers and traditional crosses, plant breeders were able to move faster and with greater precision to arrive at the desirable genotype, which results in the varieties we see in the field now.